We are all going to get started with our keynote speaker. So our keynote speaker for this year's conference is Dr. Jim Maher from the Department of Biochemistry and Molecular Biology at the Mayo Clinic in Rochester, Minnesota. Jim is a professor who leads an NIH-funded research lab working on the fundamental and applied biochemistry of the nucleic acids, DNA and RNA, as well as cancer biology. Jim got his PhD from the University of Wisconsin-Madison and did his postdoctoral training in chemistry and molecular biology at Caltech in Pasadena, California. He has 30 years of experience training undergraduate, postbaccalaureate, PhD, and MD PhD students, and is a former dean of the Mayo Clinic Graduate School of Biomedical Sciences. For many years, he has also led 2 NIH grants for the biomedical research training of students from backgrounds underrepresented in science. Jim has mentored 19 students to the PhD degree with 4 more in progress. He serves on the editorial board of the Journal of Nucleic Acids Research and is an elected fellow of the American Association for the Advancement of Science. Jim's lab has published more than 150 scientific papers, and he has been recognized 5 times by Mayo Graduate School as a teacher or mentor of the year. In addition to presenting our keynote lecture this morning, Jim will also be hosting one of the breakout sessions this afternoon, where he will lead a discussion on biomedical career path planning for undergrads and their advisors. Dr. Maher is leaving plenty of time after his keynote lecture this morning for questions and discussions, so please feel free to post your questions to the chat at any time during or after the lecture, or to unmute after his presentation so that you can ask questions. And he's asking that you don't be shy, okay? So right now I'm going to hand over to Dr. Maher, and yeah.
Oh, thank you, Dr. Thomas. Thanks, Ms. Fowler. I'm super happy to be here. Students, faculty, super proud of you as members of the Winchell Symposium this year. I'm going to share my screen and make sure that's working, and then we can have a couple comments about the process today. So maybe you can see this screen and my laser pointer. Good. So some of you are going to be thinking about your scientific presentations that start in a few minutes. Some of you are going to be thinking about judging and all that. But I'm going to challenge you to listen a little bit to the story, and then I really do want you to participate with some questions. If things come to mind, you can put them into the chat if you want to during the course of the presentation, or you can unmute your microphone later on and you can actually say things out loud. But I would love this to be a little bit interactive because the story is intended to be fun and interesting. This slide is to remind me that I've chosen a title that's supposed to capture your interest. What the heck am I talking about? Dangers of driving with a broken engine. And I hope that by the end of the presentation today and the Q&A, you'll understand why I chose that title and maybe why the particular topic today is so perplexing. But before I mention the science, I just want to tell you how excited I am to talk to undergraduates and their advisors. And the reason is that although I work mostly with graduate students and postdocs now, my own undergraduate experience was life-changing. And your careers right now as undergraduates are so exciting because of this decision you're making about whether you want to be a biomedical health professional. Do you want to be a scientist? Do you want to be a chemist? Do you want to use physical sciences? All of these things are swirling in your mind. And when I began my undergraduate career at Wisconsin, I was telling everyone I was definitely going to be an MD PhD student. And I think, honestly, I said that because it sounded really impressive. And my grandparents thought that sounded pretty amazing if you're going to do both degrees. And that's a lousy reason to choose a path because it sounds impressive. So what I want to talk to you about today a little bit in this talk, but also at the session at noon, is what are good reasons to choose the path as opposed to just reasons that maybe sound impressive to other people. So it's going to be interesting to talk about that at noon. So that's an ad if you want to come to the noon session. There's wonderful breakout topics. And so I absolutely want you to think carefully about which one you want to participate in. But why undergraduates and how they should sort of sort out the issues that they face between these different career paths is one of our topics. And when I was a sophomore and junior at Wisconsin, I had a chance to work in a research laboratory, and it changed my life because I really could understand then what the life of a PhD scientist was like and why that life was the life for me. It's not for everybody. And it's no knock on physicians or physician scientists, but for me, that was crucial. So a word or two about Mayo, and I'll also mention that on several of my slides, you'll see QR codes like this one here. And if you have a smartphone, which I know you do, you can just aim it at this and it'll give you the link to various websites. This particular one is a website for the graduate school programs at Mayo Clinic. And if you're like me, I grew up not far from Minnesota, but I had no clue that Mayo Clinic had a university environment along with being a hospital and a clinic. It's probably the best healthcare on the planet. And that sort of casts a shadow over everything else Mayo does. But there are scientists like me at Mayo Clinic who are doing basic fundamental work. There are also many translational scientists. And we have a PhD training program, a graduate school, where there are almost 200 students from all over the world that come to 3 different campuses. So there's PhD training going on at Mayo, and I want people to be aware of that. And we also have programs I'll talk about later for post-baccalaureate students, sort of gap year students. And several of those programs might be of great interest to students here. So that's my way of saying that as a biochemist and molecular biologist at Mayo Clinic, this is actually a place where we do university-style training on top of serving 1 million patients a year from all over the world. The dangers of driving with a broken engine. What's going on with that story? This is my research group. It's not so different from research groups that you may have experienced during your undergraduate years. This is a graduate program for training, and there are several adult professional scientists in this picture. But I think I want you to be reminded that even at a place like Mayo Clinic, the vast majority of the cutting edge research is being done by apprentices. And most of these folks are just a little bit older than the students on this call. And I want you to be reassured that that's where the excitement of biomedical research is, that very young apprentices are doing the essential work themselves. And so several of these people are gap year students who are doing a year or 2 of research after the undergrad was done while they're trying to decide what career they really want. And we'll talk about why I think that's great. But there's MD, PhD student in this picture, a medical doctor, and several PhD students in my laboratory. So really exciting environment, I think. And I'll talk about work that has attracted the attention of several of these students. Although my lab has 3 very different research interests, I'm only going to talk about one of them today, and it's work that some of these students are working on. Very simple outline for the next 35 minutes or so, and then there'll be plenty of time for questions. So be thinking about things that you'd like to ask about or that you find confusing or ideas you might have to help me, actually. 3 simple points for today. The first one is a why question. I think it's important always to start with why. Why do I study this weird cancer called paraganglioma? It doesn't really fit into the themes of the other things we do in my laboratory. So why in the world did I get started working on this in the first place? The second is a simple question. What are the hypotheses that we are studying to explain the origin of this cancer? I think those of us who study cancer to try to find solutions want to start with trying to understand the cancer, and then maybe we can fix it. And then the last question is, how would we dream about using basic science ideas to maybe come up with therapy for an unusual kind of cancer? So that's the outline. We'll talk about those things for the next few minutes. This is where the story starts. And I want to emphasize that many of you who are interested in science or maybe biomedical science, I know are interested in it because of your own experiences in healthcare, a family member, or your own. This is a picture from 1975 of a 14-year-old male, and it's me. This was my freshman in high school experience, which was a large abdominal surgery to remove a big tumor from the retroperitoneum, which is the area behind the peritoneal cavity. It took a lot of surgery. It was a big problem. I never detected this cancer because it was making me sick. I could actually feel a swelling in my belly and could see a pulse as if there was something growing there. And so although I didn't have any of their symptoms, I mentioned it to my doctor. And within 2 days, I'd had this big operation. And the cancer they discovered was a big tumor called paraganglioma, which I had never heard of before. And I dare say I doubt that you have. And this required then chemotherapy and radiation therapy. And I think my parents were desperately frightened that I wasn't going to survive. And this was the beginning of high school. So you can imagine the turmoil for me as a young person. But I recovered from this. And I think in the back of my mind for the rest of high school and maybe the beginning of college, I was like, well, this was so interesting. I wonder what kind of cancer this is. It made me interested about a medical career. My own experience with chemotherapy made me wonder how drugs worked, all that kind of stuff. And so it was a very personal curiosity experience, I think, that launched me into being interested in molecular biology. But honestly, I kind of forgot about it. And I went to my undergrad and I went to graduate school. I did my postdoctoral fellowship, began my career as a professor when I was 30, and found myself working at Mayo Clinic and starting here in Rochester, Minnesota. And the first thing they did was to give me a physical, because that's what they do at Mayo Clinic, because they can scan just about anything. And this was the shocking surprise in 1995, 20 years after my original surgery, almost 20 years to the week. Without any symptoms, they scanned and found that I had metastases of paraganglioma, which means the tumor had spread into my skeletons. This is my skull, and you can see where it doesn't belong, this tumor growing in the skull, and you can see it from two different positions by magnetic resonance imaging. It was a big shock, because when you're a cancer patient and you're told that your cancer has spread to your skeleton, that usually is a very, very dire circumstance, because it's much harder to treat cancer that's spread in this way. But I'll have you notice that was 1995, 25 years ago. And so I'm going to be telling you the story of being a cancer patient for years and years, not months and months, and how that's allowed me to be interested in the biochemistry of this cancer. But it's a cancer that can start and it can spread years later, which was fascinating. Look, that was one spot in the skeleton. 33 years later, by 2008, this is my hip. And you can see what the hip is supposed to look like, the pelvis. And here is a metastatic lesion of paraganglioma growing in the bone. And you can see it as two different sections. So the magnetic resonance imaging technology lets you see where cancer is spreading in the body. No symptoms, no pain from this cancer. It's spreading sort of innocently, but potentially doing damage to the skeleton. So it was important for me to have that monitored and eventually also treated with radiation. By 2016, that spot that had been radiated had grown back, the metastasis in the iliac bone. You can see this is a PET scan, positron emission tomogram, where fluorodeoxyglucose is what's being detected. And when this is injected into the body, tissues that take up lots of sugar are really good at sucking it out of the blood, light up. And if it doesn't take a minute to see one of the tissues that loves sugar, it was the brain. The brain lives on sugar, and so it lights up in a PET scan. But here is a spot where there shouldn't have been sugar, which was the metastatic lesion of this cancer growing in my pelvis. There's a little bit of red in the kidney and the bladder just because the fluorodeoxyglucose is being excreted, and that's not cancer. So this had been radiated, so we couldn't radiate it again. So another technology to treat cancer was brought, and Mayo does this kind of amazing stuff. This is a close-up of my pelvic bone, and this is where the bone has been destroyed by the action of the metastatic cancer. This is a skewer that's been placed, luckily under anesthesia, deep into the bone, and 3 or 4 of these skewers are placed in by an interventional radiologist, and then they freeze that spot with high-pressure gas in these needles that chills that tissue, and they freeze it and thaw it and freeze it and thaw it 3 or 4 times over the course of a half an hour. And that freeze thaw is happening in this dark region, and that can kill tumor cells, just like freeze thaw of cells you might do in a laboratory can lyse them and kill them. So this is, instead of radiation or chemotherapy, literally putting skewers in and freezing and thawing the spot called cryoablation in the bone. And it's kind of amazing. I'm glad I wasn't awake for it, but this was the day before that procedure was done. And here's a PET scan showing all the uptake in the tumor. Again, bladder and kidney uptake is just part of excretion, so not to worry. But this is the day after that procedure, and it just shows how efficient and accurate the diagnostic or the interventional radiologist was, who could freeze and thaw that and kill the tumor without using drugs. So my experience as a cancer patient has included radiation and drugs and surgeries and another set of radiation, but then even techniques like this one, which have to do with freeze thaw, right? So I have survived with paraganglioma, metastatic paraganglioma, for years. And I'm deeply thankful for having not only a tumor that's slow growing and I living with it rather than dying from it, but also having an excellent healthcare team. So why I study this cancer is not so much because I need to cure myself, although there are many patients, sadly, with paraganglioma who can die from the cancer, and we need to try to find therapy for them. I got interested in the paraganglioma for the reasons I'm going to describe in the next few slides. It's because of how bizarre the biochemistry is. And because I'm a biochemist, my curiosity just said, well, I wonder if many people are working on this problem since it's a rare tumor. And my doctor said, not really. If you are interested in trying to figure this out, there's no rule that says you shouldn't be able to work on it. And that's another beautiful thing about being a PhD scientist. There aren't rules that say what you can and can't study. You simply have to find money and you can organize a team to study a problem. So why I study this is my curiosity about my own cancer. And I'll talk a little bit now about the hypotheses we're trying to resolve about what's going on in these cancers. What is this cancer? Paraganglioma. It's rare. There's only about 1 out of every 300,000 people in person years that have an incidence of this tumor. So you probably won't meet another person like me anytime soon. Every cancer is some kind of cell growing out of control. And the cells that are growing out of control in my cancer are called neuroendocrine cells. They're cells that receive a nerve impulse and then they secrete a hormone in response. So they're sort of a cell that bridges between the nervous system and the endocrine system. If this kind of tumor happens in the adrenal gland, it's called a pheochromocytoma, which is confusing, but it's just a paraganglioma in a different place. But otherwise, tumors like mine can occur from the pelvic floor to the head. Some of them cause high blood pressure because if these cells are growing out of control and are confused, they may do what they normally do, is to secrete hormones like adrenaline. And if this tumor is occasionally secreting adrenaline without any logic, it can cause problems like high blood pressure. Some of them, like mine, are malignant and they spread and they can kill you. And here's the really interesting part that caught my attention. Something like 30%, maybe more, run in families. The risk for paraganglioma is inherited from parents to children. And whenever you hear about cancers that run in families, that tells you that there might be a clue to the genetics right there. And that might be a fascinating way of understanding what's going wrong. So hereditary paraganglioma, which is paraganglioma that runs in families, is what I have. And I have other members of my family who are at risk, but I'm the only one that's had big tumors so far. And here's what got my attention. When I learned about this cancer, the physicians told me, you can read about it, but the mutations that you inherited that put you at risk inactivate this enzyme, succinate dehydrogenase. And I think when I first heard this, I argued with my doctor because I'm a biochemist. And he said, yeah, I think they said the enzyme that's deleted in these cells is succinate dehydrogenase. And I said, that does not make any sense, because some of you may recognize that's an enzyme in the TCA cycle, the tricarboxylic acid cycle, the Krebs cycle, right in the core of the energy generation system of the cell. It's an enzyme in the mitochondrial inner membrane in the mitochondria. It's an enzyme that's in the engine of the cell, and it's broken in my cancer cells. And I said, that is bizarre. Why would breaking one of the key enzymes that drives energy production from sugar cause confusion in a cell as opposed to just killing the cell? So you can see that as a biochemist, this doesn't make sense. And I'm like, that's weird. In my case, every cell in my body has one good copy of the chromosome that carries succinate dehydrogenase, this particular subunit. And I inherited a broken one from my father. So every single cell in my body is heterozygous for this mutation. The cell doesn't have any problem if it has at least one good copy, but occasionally, just by rare errors, the second copy is lost. And now we have a cell with no copy of this succinate dehydrogenase machine. And this is oncogenic. It causes cancer if the cell has no copies of this, which is really interesting. And what's even more interesting is that that second hit, the loss of the other copy, is only tumorigenic if it occurs in neuroendocrine cells. So every cell in my body looks like this on the left. But if I lose the second copy in a blood cell or a skin cell or bone cell, it doesn't cause cancer. It's only carcinogenic if that second thing and confusion occurs in a neuroendocrine cell. That's also bizarre. So this is a bizarre cancer. It's bizarre because we don't know why breaking the Krebs cycle causes it. And it's bizarre because we don't know why it only causes that confusion if it happens in a neuroendocrine cell and not other cell types. This idea that genetic changes can be carcinogenic in just one tissue and not another is known for other cancers, and BRCA1 and 2 mutations that you hear about that can cause breast and ovarian cancer similarly occur in every cell of the body, but the female in that case is only at risk for those kinds of cancers. So that's bizarre. So a review of cancer biology and what you may have heard about in thinking about why cancers form and then why we're studying this weird one. You can think of cancer like a car that's driving out of control, and that's dangerous, right? So if we think of the analogy of what's wrong with a car and what's a corresponding example in the cell and what the corresponding mechanism is, maybe you've heard of some of these before. If your car can't stop because the brakes are broken, the equivalent example in cell biology is if something like the p53 tumor suppressor gene, which is a recessive tumor suppressor, meaning you need to lose both of them, both mom and dad's copy before you have a problem. If that's broken, you can't stop, just like brakes don't work. And it's obviously a more elegant problem in a cell, but that's brakes not working. What about if you can't unaccelerate? The other obvious problem is if your car has its accelerator pedal stuck to the floor and it's floored all the time, you can't slow down. And the positive example here is the RAS dominant oncogene mutation, where all you need is one broken RAS gene and it's flooring the accelerator all the time and the car is going too fast. So these are cancer mutations that are unfortunately common, and it's sort of easy to understand what the problem is in those two cases. Here's what attracted my attention to paraganglioma. This car has an engine defect. That's weird. The succinate dehydrogenase enzyme is broken, and it's acting as if it's a recessive tumor suppressor like p53, which doesn't make any sense. And we have no clue going into this what the mechanism is for why breaking something in the engine would cause a cell to go out of control somehow. So let's talk about that. What is it that's broken in paraganglioma tumor cells? And for those scientists who are studying this weird cancer, what is their hypothesis that they're trying to understand or test to see what is going on in this cancer? Because if we can understand what's broken, maybe we could understand how to fix it, or maybe we could find a vulnerability that would kill these cancers faster than other cells around them. And here's the machine that's broken. It's called succinate dehydrogenase. It's an ancient enzyme. It's present in bacteria and fungi. It's present in mammals. It's in plants. It's just an ancient part of metabolism. It's just the inner mitochondrial membrane, and it's got 4 subunits. This is an X-ray crystal structure, so we know all about the atomic detail. A, B, C, and D are the 4 subunits that have to assemble to do this job. It has a prosthetic group, FAD. It has 3 beautiful iron sulfur clusters that carry electrons from the substrate into the membrane where they're given to coenzyme Q. And electron flow, you'll remember, is what the central part of making ATP is all about. So defects in any one of these 4 proteins, they're all encoded by genes in the nucleus, but get assembled in the mitochondria. Any defects in these 4 can predispose patients like me to paraganglioma. So something about this machine, when broken, confuses cells into growing out of control. Now, although this thing doesn't actually spin in the membrane, I'm just rotating it so you can see how elegant it is. This is FAD, and this is where succinate binds here in the catalytic subunit. The electrons flow through these different iron sulfur clusters into the membrane. And so when things are broken, like in my cells, my cancer cells, the B subunit is gone. So the machine can't do its job since there's no way to shuttle electrons into the membrane, and we can't generate any electrons from this, and we break the TCA cycle at this step. And I'll show you what I mean right here. When I was your age, I had to memorize this. So if you just get to Google it, times have changed. But in my generation, we had to know that glycolysis, which is up at 12 o'clock, brings acetyl-CoA from splitting sugar. And then we go clockwise around the Krebs cycle. changing from one intermediate to another during metabolism. And as we do that, you'll remember the point is to oxidize or remove electrons from these intermediates so that we can use those electrons to build ATP by making a gradient of protons in the outside versus inside of the mitochondria. This is the situation where we are trying to do this in the mitochondria. And this is the step that's broken in my tumor. Succinate to fumarate, simply changing a single bond to a double bond by taking off a pair of electrons. When that's missing, all of the steps that are downstream are broken and the Krebs cycle is not working anymore. So we don't have the ability to populate these intermediates because we're not continuing past this important step. Very bizarre that this would be something a cell could survive and that it's something that would cause cancer. So why or how might losing this enzyme of the Krebs cycle confuse cells? This has been haunting me and I think other researchers in this field ever since I first heard that it was an SDH mutation. What could be going on? Maybe you can think about what weird things you could imagine might happen when the cell's in this situation. For one thing, it's not generating as much ATP anymore, so I can't see why that would confuse it, but the main hypothesis is this. It's called oncometabolite accumulation. In the chemistry of the mitochondrion, when you can't get past that block, everything upstream can accumulate, and these normal metabolites can become oncometabolites, meaning cancer-causing metabolites, and the question is how? But here's the picture, and here's what I want you to think about. If we put a block right here in a cycle, we can't continue, so that's part of the problem. But the other problem is that the metabolite intermediates upstream accumulate, they literally back up like blocking the flow of a pipe. When you push liquid through the pipe and you can't continue it, you're going to fill it up with these intermediates. And here are the two most proximal intermediates: succinate, the precursor, a friendly dicarboxylic acid, and succinyl-CoA, which is a high energy form of succinate. So our hypothesis we're studying is that it's the accumulation of these two friendly molecules that's causing trouble. These are the oncometabolites we're trying to study. Here's succinate, very simple molecule, and here's the full structure of succinyl-CoA. If you've never seen the coenzyme A part of this chemical, it looks a little bit like adenine or adenine diphosphate with another phosphate here. This is the high energy form of succinate called succinyl-CoA, and these two things are accumulating in these tumor cells. And what might that cause as a problem? Tell you for a couple slides what we think succinate does and a little bit about what succinyl-CoA does, and this is what our lab is studying. Succinate poisons a whole class of enzymes called dioxygenases. Dioxygenases take some substrate and they split oxygen in the presence of iron, and they do something to the substrate, like often adding one of the oxygens to that thing. And this can be a protein or it can be a nucleic acid of different kinds. So there's a variety of different dioxygenases. But all of them have this weird chemistry where alpha ketoglutarate is one of the co-substrates for the reaction, and they produce succinate and carbon dioxide in doing this job. So succinate fits in the active site of this enzyme since it's one of the products. And it turns out if succinate is very high in concentration, like inappropriately high, like in my cancer, it'll bind competitively in the active site, and these enzymes, dioxygenases, get inhibited. Intriguing. So what are these enzymes and who cares about them? Well, one family are called prolyl hydroxylases that take oxygen and they add one of the oxygens to a proline. The amino acid proline here gets hydroxylated. And that's a signal that won the Nobel Prize last year to understand that this signal is very important for deciding whether there is or isn't enough oxygen in a cell. And prolyl hydroxylases can't do this job if there's too much succinate. So that's intriguing. Here's another enzyme in that family, the histone demethylases. This is an imaginary histone with a lysine amino acid side chain, which has been methylated, trimethylated, which is one of the histone code marks in epigenetics in the nucleus. You need enzymes of this family to remove those methyl groups to control gene expression. And those enzymes are poisoned by succinate also, which means that's a potential problem in the nucleus. Here's another place where those enzymes are important. If you have cytosine with a methyl group, the first step to removing the methyl group, which may be important for gene regulation, are cytosine demethylases, and they have the same chemistry. So too much succinate has this unexpected effect of poisoning a bunch of enzymes that are important for controlling what proteins are alive and whether the nuclear gene expression is proper. Isn't that interesting? So we have a problem in the mitochondria spilling out, causing confusion in the cytoplasm and confusion in the nucleus, which is unexpected and maybe something that we'd have to do something about. So this is a slightly busy slide maybe, but it just summarizes that in the mitochondria, we normally are doing the Krebs cycle here. And in the cytoplasm, we normally have enzymes that are using oxygen to mark proteins for destruction. And in the nucleus down below here, we have enzymes that are normally regulating whether histones are methylated or whether DNA is methylated in the chromatin. And in my tumors, all these things in red are going on. Succinate is accumulating and acting as a poison, a competitive inhibitor that blocks this family of enzymes in the cytoplasm, which allows some proteins that should have been degraded to go into the nucleus and turn on gene expression. Big confusion. And those succinates also can do things here in the nucleus, like confuse which histones are supposed to be demethylated and which DNA is supposed to be demethylated. So that's a lot of problems from one enzyme difficulty that's happening in the mitochondria. So that's succinate accumulating and causing a problem. What about succinyl-CoA, this high energy form? It's also capable of causing problems, and we're just studying this in our laboratory now. It was something we discovered. Succinyl-CoA is a reactive metabolite, and it can succinylate lysines on proteins that have lysine side chains. And here's what I mean by that. Here's a lysine hanging off of a protein. You remember this side chain from chemistry and biology class. It's a cation, NH3 plus in the side chain at normal pH, and that doesn't react particularly well with succinyl-CoA. But there's always an equilibrium, even at pH 7, between a part of the lysine that is going to be neutral, and this is now a good nucleophile. It has a free pair of electrons, and it's very good at attacking electrophiles like this high energy thioester bond. And succinyl-CoA will succinylate lysine side chains if there's too much of this floating around the cell. And look what happens. We've turned a positively charged amino acid on a protein into a negatively charged amino acid. We flipped its charge from +1 to -1, and we've added this bulk. So you could imagine that succinylating proteins could really change their behavior. They could change the behavior of an enzyme. It could change the folding of a protein. So this poisoning of proteins with succinyl-CoA accumulation is something that we're also studying. So in this analogy of what's wrong in the cancer car of paraganglioma, the best analogy I can come up with is that breaking the engine is actually creating fumes, like improperly burned fuel, that can intoxicate the driver. So not like these easier to understand situations. It's the metabolites that accumulate that are causing the problem, and we're trying to understand exactly how and what you might do about it. So we've talked about why I study this. We've talked about some current ideas for what might be going wrong, and I think the obvious question for all of you is like, what could you do about that? You have a broken gene. I suppose you could try to replace the gene. So gene therapy might be used, but you'd have to get that into every cell that's a cancer cell if you wanted to correct them all. So what are we going to do about it? Here's a question that I asked with one graduate student some years ago, and then a series of post-bac students. These are gap year students, and they all happen to be from St. Olaf College. And it's not that I only take St. Olaf students, but St. Olaf isn't far from Rochester, and we have a wonderful connection. So these students, 1 or 2 years each, have been working on this problem, and we are asking the question, can we suppress or heal the effects of these SDH defects, and then by using that process to treat SDH lost cells. So tumor cells that lack SDH are accumulating things like succinate and succinyl-CoA. Can we find ways to prevent that accumulation? Would that be a useful drug therapy? So Emily did her graduate work here. Megan, who's now a neuroscience student at Wisconsin. Caleb, who's now a physician's assistant applicant in Omaha. And Williams, who's in my lab right now, are all brilliant students who've been working on this problem. And they've been taking advantage of the fact that we have a huge obstacle in this field, that there are no paraganglioma tumors that form in animals. So we have no model like mice that get paraganglioma, and we have no paraganglioma tumors that grow in dishes. So this is a huge obstacle, because most cancer scientists can study their tumor by studying mice that get the tumor, or they can study the tumor because they have cell cultures of the tumor growing and they can study what to do with it. We don't have that stuff. This is a weird tumor and it doesn't form in animals and it doesn't grow in dishes. So what have we been studying? We've been studying a yeast model. Yeast is not a cancer, but yeast, Saccharomyces cerevisiae, budding yeast here, we study for other reasons in our laboratory, and they're a eukaryote, and they have succinate dehydrogenase, and they have a Krebs cycle, and they have a nucleus. They're like little humans, except they're all identical twins, and you can grow trillions of them in beakers very quickly. And we can then study yeast as models for what's going on when we have succinate dehydrogenase problem. And succinate dehydrogenase, when you delete those genes from yeast, they can only grow by glycolysis because their Krebs cycle also doesn't work, just like my cancer cells. And you can see that because if you take the yeast and just plate dilutions of them, a medium amount, and just a few cells on a plate with glucose, they grow fine whether they have SDH or not. But if you grow those same yeast on a plate where the only carbon source is glycerol, they can't burn glycerol. They can't use the fermentation, glycolysis works glycerol, but they can't burn it. That's what I'm trying to say. And you can see that the normal yeast do fine because they can burn the glycerol, but the yeast lacking SDH barely grow at all because they don't know what to do with this carbon source since they don't have a Krebs cycle. So the cells are disabled, kind of like the tumor cells are. So it means we can study them. And let me just show you the trick we've been using in the last couple minutes, what that group of students have been trying to study with yeast. Yeast cells have a mitochondrion like we do. They have a nucleus, all the same kinds of things that we have. But in their cytoplasm, they have a special enzyme called JLP1 that's one of these dioxygenases that's vulnerable to being intoxicated by succinate. And we realized that this enzyme might be a useful enzyme to learn about how to help yeast that are accumulating succinate, because in yeast, this enzyme is needed if we feed them only sulfur form that is in this sulfonate form, and they need to convert it to sulfate or sulfite. Yeast need to get sulfur for amino acids, and they're struggling. If we only give them sulfonates, they need this enzyme in order to convert sulfur into a form they can use. And if the cell is being flooded with succinate because of this SDH problem, if we make the yeast like a model of our tumor, then the succinate poisons this JLP enzyme like everything else, and the yeast can't grow on sulfonates. They can't convert this into a useful sulfur source. And so we have a model that yeast that have the SDH problem, not only can they not grow on substrates that require TCA cycle, they can't even grow on sources of sulfur with this form because they can't convert it. And that gives us an interesting tool for looking for things that would overcome this problem, because we just have yeast growing or not growing, and that's an assay you can measure. So the idea these students have been asking is, let's take 1,280 known drugs that are already approved for human use, and let's study them one at a time in a little growth assay and look for compounds that improve the ability of SDH loss yeast to grow on this sulfonate, this stuff, as the only sulfur source. In other words, the only way they can grow on this is if this enzyme is working, but the enzyme is being poisoned by succinate. So we're looking for anything that could rescue the yeast from the effects of succinate accumulation. And this is a screen, which means we're not telling the yeast how to do this or telling the drug how to do it. We're just asking, does any drug know how to solve this problem? And if we can find one, then we can ask how it works later on. And yeah, I welcome comments or questions in the chat, and we'll come back to those in just a second. So keep them coming. So we're going to ask for, are there any drugs in this library of 1280 that can rescue the ability of these SDH loss yeast to grow on this sulfur source, which implies that this enzyme that's been poisoned might be rescued. So the students ranked all the drugs after doing this growth assay in microtiter plates. We ranked them from 1 to 1200 on the basis of how well they help the yeast recover. So drugs that hurt the yeast or make it worse have low scores on this chart, and drugs that do well will have high scores. And so when we ranked all of those drugs after growing the yeast in the presence of 10 micromolar of each of the drugs, there were a group of compounds that helped the yeast do better. You can see a lot of them made the yeast sicker, which is scarcely a drug in this case. Again, we're not trying to kill the yeast, we're trying to rescue them to show that we can suppress this problem. But these active compounds up here help the yeast grow better in this assay. And then we can start saying, who are those drugs? And eventually, by looking at them, we might be able to figure out what they might be doing to help the yeast do better and why they're rescuing that enzyme. So just looking at these cool drugs, you couldn't guess what might be going on with them. But I think it is interesting that 3 of the drugs that help the yeast are drugs that can chelate iron, suggesting that some of the problems that are causing this enzyme not to be able to work when we're accumulating succinate are caused by things that could improve if we chelate extra iron in the cell, which is a surprise. That's not something we normally would do to solve a cancer problem, but it might be a direction that we could take. So the next steps in this kind of work are, well, how are these drugs that screened as being helpful in saving the life of these yeast cells that are struggling because their JLP1 enzyme being intoxicated by succinate, how do they work? Can we use this yeast model, or can we find better models like other kinds of cells, or eventually can we make mice that get this kind of cancer so we can study it better? And then can we use these basic science ideas of too much succinate, too much succinyl-CoA to generate some kind of a therapy that might take advantage of the fact that this kind of tumor is really different from a lot of cancers because it has this fundamental metabolic problem. And maybe we could address the metabolic problem and try to rescue the cells rather than just trying to kill them. I think many cancers are just about kill the cancer. And I have this dream that maybe, since the problem here is a metabolism problem, that we'll be able to find drugs that change the metabolism or unconfuse the cells one way or the other. So what I wanted to talk about today then is why I got interested in studying a rare cancer, even though my lab usually works on nucleic acids. I try to show you what our current hypotheses are that we're testing with other labs around the world to say what's really the fundamental confusion in these cancers. And then the idea that maybe we could find some kind of therapy based on the biology that we've learned from the yeast cells. This QR code, if you aim your phone at it, is just a link to my own laboratory website where there's a description of our lab, our students, what the students are working on, and as well as the other projects we do in my lab, just so you can see this cancer work in the context of other work. And here's all these great students that have worked in the laboratory on this project over recent years, including several gap year students. And at the noon session, I'll talk a little bit about why many of you are more and more attracted to gap year post-baccalaureate studies and what I try to accomplish during those years. Then we have collaborators, and the funding for this came from the NIH and particularly from the Paradifference Foundation, which funds research on rare cancers like paraganglioma. All right, so thank you very much for your attention. We have a few minutes, and I know our sessions start after 10 o'clock, and I wanted to leave time. And so there are a few points that are already in the chat we can look at, but I would welcome folks after I answer those to just open mic and ask questions. And you're welcome, if you're a student, you're welcome to ask questions. If you're a faculty member, I know the students are all like, I gotta get ready for my presentation, but relax, we'll do this sort of till the top of the hour, and then we have time for you to shuffle and regroup and come to your session afterwards. So let's look at a couple of these. I'm going to leave the slides up for a second just because there might be some pictures here that are useful, but in a minute, if they're not, we'll skip them. So Felicia was asking, what was the process to choose Saccharomyces cerevisiae? And 2 points to Felicia for spelling that right in the chat. I would not have. as a model system? Are there limitations to working with it for your work now? So it's clearly weird to be working on a yeast when you're trying to study a human cancer, because a yeast cell is not a cancer cell, and yeast cells don't have some of the equipment that a cancer cell will have. But my attitude has been, I wonder if there are fundamental things we can learn about metabolism from a simpler organism that has some of the same machinery that we have. So these are overarching hints. And if we found something in yeast, like, wow, a chelator seems to help this problem get better, we could start saying, well, I wonder when we start having models that are better in mice or humans, whether we could use that idea to test it. So they're giving us hints. I don't know that I would take a drug because it helped a yeast cell. That would be like, that would not be a legitimate reason to move something directly to the clinic. But because we don't have good mammalian models, we don't have a lot of choices. And so our argument is finding something like a eukaryote sister to ours, like yeast, might be able to give us an insight. It's certainly a lot cheaper to study, and every single yeast gene is known. The entire organism is sequenced was long before humans, and you can actually procure every single knockout yeast cell from a library so that you can study the behavior of every individual yeast gene knocked out, and that strain is easy to get. So it's so much easier to study the biology of these questions. You don't need CRISPR-Cas9, you don't have to build gene knockouts or deletions, they're already available. And so all of these things are faster and easier, and this is a wonderful microorganism that's been studied for generations. So although it's not cancer, it has huge advantages that that we want to take advantage of when we can. So I'm just going to go back here so you can appreciate its beauty. It's, again, in this case, the budding yeast that we're talking about that's easy to grow. So Joanna is asking, do you think the microbiome could influence this? Some bacteria are known to chelate iron. Great question. We don't know what features of the patient make the tumor grow worse or better, given that the tumor has this metabolic problem. It's a reasonable question to say, are there things that we could do that might change the metabolism of the tumor or might change its environment? For example, the metals around it. So if we have models, maybe we start with yeast and say, which of these chelators can we just add to the environment around the yeast to change them? I suppose you could make a model, Joanna, where you actually co-incubate the yeast with some friendly bacteria in a mixed culture and ask, does the environment around the yeast cell change this confusion? But what we really would want to do is what we would do if we had a breast cancer to study or a colon cancer, where we have mice that can get those cancers because we have genetic models for mice that give them the same cancers that we get, which that's not available in paraganglioma. If we had that, then you could ask all these questions. Well, let's grow the mice in a germ-free environment where they have no microbiome at all. They're sterile. And you can do that with mice where they literally don't have any germs living with them. It's kind of cool to me that they do fine. We like to think the microbiome is a live or die situation, but you can grow animals in sterile conditions with zero microbiome. You can also grow them and change their microbiome or delete their microbiome and repopulate it. And then we would be able to ask the question you're getting at, do any of those changes have impact for how the tumor grows? We'd love to do that, because if you could show that changing the microbiome in a patient has an outcome for their biology, that's not a toxic thing to do, right? And we, as you know, don't know enough about that. That's an example where I think doing this with mice, if you have a tumor model in the animal, is the perfect solution. Again, mice aren't humans, but to study the microbiome in a rare cancer like this, there just aren't enough patients to do a controlled study where you have very similar patients and some get one therapy, some get the other. It's called a controlled double-blind clinical trial of the microbiome. So hard to do if it's a rare cancer. So much easier if you have dozens of animals who are identical twins, or trillions of yeast cells that are identical twins, because then the genetics of the patient are not part of the question. So I think it's a great a great point, Joanna. Thank you. And let me know if you want to follow up at any point. So Angela may have said this. I was excited about everything that was, and was taking a lot of notes. Did you say what your mutation in SDH is? Point mutation, expression levels. Yes, and Keenan is one of my very positive and fun current PhD students working on a totally different project, but yeah, Keenan's a Bethel graduate. In my case, the mutation I inherited from my father is a splice donor mutation in the SDHB gene. So you asked, but I think this is where you drill down and say, well, what's actually broken in my chromosome? There's a point mutation at the place where normally one of the intron exon junctions needs to be read by the spliceosome to remove that intron and allow two exons to be assembled in SDHB, which is that middle subunit that has the iron sulfur clusters. So in my case, because of that point mutation, that splicing is not properly done and an intron gets left in. And if you leave an intron in a message, I think you can imagine that the exon coding sequence doesn't make any sense anymore, and you finish an exon, and instead of moving on to the next one, you read into the intron sequence, which is gibberish, and you eventually will just hit by accident a stop codon. So in my body, the SDHB protein that's encoded by my dad's chromosome just makes a little bit of the protein and it gets to a stop codon. And so this whole question that Angela's raising, which is what the exact mutation that the patient has, will dictate whether they have no protein if they lose the other copy, or if they have a small amount of protein. And maybe that dose of the protein has something to do with how serious the cancer is, because maybe it dictates how much succinate or succinyl-CoA accumulates, right? So great question. You can imagine the genetic counselors and the geneticists who discover that families have SDH mutations need to be thinking about, well, what's the particular mutation? Is it a mutation that's very serious? What about mutations where we can't really tell? I think that's a fascinating question. Let's say it's a mutation that changes isoleucine to a leucine amino acid. That's a subtle change in biochemistry. Is that enough to really cause a problem or not? And so if that patient sequence comes back, we have to figure out, well, is that a serious mutation or not? And you can imagine for other cancer problems like BRCA1 and BRCA2, knowing whether the mutation you carry is a cancer risk or not is a huge issue, because it may determine whether you have prophylactic bilateral mastectomy and have a big surgery to remove internal organs to save your life. And you don't want to do that if the mutation you carry isn't serious. So it's a very insightful question. Stephanie, is it possible that other metabolites before succinyl-CoA also accumulate and cause problems? In other words, you're thinking ahead here. If you're backing up the metabolites at the previous two spots in the Krebs cycle, isn't it possible that there are problems upstream of that? In other words, this question. I made a big deal about these two metabolites being backed up, but what about accumulating these ones and these ones? So when we make these yeast models, or if we break this enzyme in tissue culture cells, which also aren't tumors, we can ask how much of these metabolites are present by breaking the cells open and using metabolomics. And what we see is a lot of these two, but not much accumulation of these ones, probably because there are enzymes that feed into the Krebs cycle through alpha-ketoglutarate here that can allow this metabolite to be transformed into other metabolites that are not part of the Krebs cycle. And so they can relieve, if you want to think about it, they can relieve some of the pressure. But those transformations aren't available here as much. There's a little bit of an alternate path for this one. So there's not much evidence that there are problems caused by up here. Interestingly, and Stephanie, it's a great question, because there are mutations of the next enzyme in this pathway, fumarate hydratase, that also cause cancer. And there are some mutations of the enzyme that goes from here to here that don't make more alpha-ketoglutarate accumulate. They make a different version of alpha-ketoglutarate that's a cancer-causing metabolite. So your point is really insightful, that breaking the Krebs cycle in different ways we're teaching us is ways of creating too much fumarate, succinate, succinyl-CoA, or other similar metabolites that are caused by enzyme defects, not just breaking the enzyme, but changing its biology. And that reminds us that in the metabolism of the cell are potential carcinogens, if you want to think about it that way. These aren't DNA damaging agents, these are confusing agents. And the take-home message here is in the engine of our own cell, there's potential to cause problems that create fumes that may be confusing. And I think the take-home message is going to be, what is it that we might do now that we understand that to help improve the situation? So we're right close to the top of the hour. I'm glad you were able to listen for this, and thank you for those that posted questions. If you have questions that I didn't get to, you can look me up and I'd be happy to talk by email. And again, for those who are interested in sort of a perspective on choosing between biomedical and bio and other chemistry related topics, I'm happy to chat at noon about that. But super proud of all of you. I hope that you have a great time presenting during the next sessions. And thanks again for the invitation, Chris and Aeisha.